Anyway, apparently this is something that Sandy Young found. So if you don't like what I say, Susan, then you can peck on the person in front of you.
It's out there on the web.
I'm sure it is, whatever it is.
So anyway, Susan's passion for science started when she was 8 years old, when her father gave her a microscope and told her to go out and examine the world. And that she's done. Now, I think she originally thought that she was going to be an academician, like I thought I was going to be an academician too, but she decided to accept a position at Merck. And from there, she went, I'm not sure exactly every step, I'll let Susan sort of account for those things, but in 2011 she was Fierce Biotech's, one of the women in biotech. She was a CEO of a company called Proteolix that was acquired several years ago by Onyx, and is currently, her company started the development of a therapeutic that I think is going to show great promise in the area of multiple myeloma. She's a serial entrepreneur, so she's now the CEO of another company, and serial entrepreneurs are good to model your behavior after because the world has too few of them. So without any further sort of revelations about Susan's past, I'll invite Susan up to give her words of advice.
Thank you. The web is a wonderful thing. Once your data is out there.
Once you're there, you're always there.
It's true. My father was probably the primary reason I'm here. And I don't really like to talk in generalities. I'd really rather tell you something about my story. I think that's more interesting. So I will start with my father. He was the son of immigrants. He was born in the United States, but he was the first person in his family to have any kind of education, really. And he was the first person in his family to go to college. And he was the beneficiary of the GI Bill in World War II, so he was able to switch from a local college, Brooklyn College, where he was first started, to Columbia University. So he wound up going from nowhere to graduating from a very prestigious university. And he became an electrical engineer, and he was involved in research, so he did a lot of work on sonar and on aircraft simulators. And after that, he became a manager, and he worked for the federal government for many, many years. But he always really loved research. And he had me and one sister of mine, so he had two daughters. And I think he would have honestly lavished plenty of attention on sons, but he didn't have any. So my sister, as a result, has a PhD in economics, and I got my PhD in biology. So he turned his attention on us. He tried to get my sister to do a chemistry, so he bought her chemistry sets. I must have rebelled, because I never got the chemistry sets. And instead, I really did get a microscope when I was 8. And what was important was that there was no structure. He just said, this is great, you should just go do something. And so I went down to the pond that was near my house, and I put a drop of water on the slide, and I was just so thrilled that I didn't know what I was going to see, and then I saw something. And that really stuck with me. He also used to drag me out at night and make me look at the stars in a telescope, but I'm not a physicist, so I think I like the microscope better. And he made us a lot of toys, you know, that like trucks that went when you shined a flashlight on them and things like that. And the most important thing he said to me was, you can understand the world. You can understand anything around you, and as long as you work hard enough, you can understand it. So I thought that was very, very important for all the years of education that I went through subsequent to that. The other thing that he did was instill in me a love of solving problems. So I thought that was very important. I do remember one time when he came home from work because a problem had been circulating amongst the engineers. And it's probably a problem you know now because I know it's on the internet. And it's the problem where you have to weigh 3 times 12 balls, one of which is lighter or heavier than the other, and have a shortproof way to do the weighing such that you can tell which ball is lighter or heavier. And I couldn't solve the problem, and I remember, actually I teamed up with my mother, who wasn't particularly into math, And it took me 3 days to come up with the answer. But I remember that the important thing was I couldn't stop. I couldn't leave the problem alone until I'd figured it out. And I certainly didn't need, whether I was going to get an A or something on it. I just had to solve the problem. And of course, my father was super supportive when I finally came up with the answer. And my mother was there cheering me along. And it was really a feeling that never left me. When I pursue my interest in science, as I went on in my career, I just wanted to know the answer. And I walked out of my childhood believing that it was important to be passionate about what you were doing. You can find the answer to something if you work hard enough at it. And the motivation really should come from you. You don't need external motivation. You want to solve a problem, you can solve it. And so the upshot of that was you should do whatever you feel passionately, strongly interested in. And that's what I did. So yeah, my father was a scientist and he believed that science was the greatest good, and that certainly, got passed along to me. But the truth is what he really said was just do something you're passionate about. And he was very supportive of my career the entire time. He passed away a couple of years ago. But he got to see me go all the way to be a CEO. And his only comment along the way was, I hope you're not investing your own money. So he was a little more risk averse than I was, and I'm not. So that was really my first mentor, and I'll tell you a little bit about how I got to run a company and then talk about my second mentor. So I followed a pretty traditional career path. I signed up, to go to college. My way of picking a college was that I didn't want to fill out any applications I didn't have to fill out. So back then, early acceptance was a really new idea. I thought it was great. And so I just applied to Smith College, and I got in, and I took the entire box of applications that I hadn't filled out yet and threw them in the trash. So this is pre-computer, pre-rolling admissions. And that's really, there was not much more thought. That's why I went to Smith. It turned out to be a great school. It's still around. It's single sex, so it was all women. They actually have a great biology department there. And I wound up being in a whole set of classes that were for nothing but women. And at that time, because there weren't too many women in science, it really mattered to have that kind of group of people around you and an institution that existed just for women. So I came out of Smith with a biology degree, and I thought I was going to go to medical school. And I applied to medical school, and I didn't get in. And it was the first time that anything like that had ever happened to me, and I recommend very strongly that you try to fail at something. Because it's really important. I'm sure you guys are all super smart. I did very, very well in school, and everything was easy until I didn't get what I wanted. And I had to rethink, why was I applying to medical school in the first place? And along the way, not because of medical school, but because I loved this particular professor who taught embryology and development biology, I just wound up in her lab doing, on my own, a senior thesis project. And it was working in the lab by myself that I just fell in love with, but I didn't know that before I did it. And I just loved being in the lab by myself, doing my own thing. I liked the actual work. Nothing I did my senior year revolutionized development biology, that's for sure, but I just loved completing a project. And I realized when I didn't get into medical school, maybe I should just take some time in a research lab and see how I feel about it. So that's what I did, and I went actually to the USDA. They do have labs. I went to a nutrition lab for a year. And it was a very sobering experience because their view of science is not my view of science. It was very, very laissez-faire, and the bar was really low. And I thought, I can't ever be in a place like that. And so I turned around and wound up going to Johns Hopkins University for graduate school, where the bar was very high, and where I was much happier. So I went to Johns Hopkins and got a degree in a PhD in genetics. And I worked on a virus called Visnavirus about 3 years before AIDS became nailed down as a retrovirus. And it turned out that Visnavirus was a neurovirus, so it hit the CNS, the central nervous system of sheep, and it was a good model for multiple sclerosis. But probably more importantly in the long run, it turned out to be a great model for AIDS. And so when the HIV virus was nailed down as the genomic source of AIDS disease, Visnavirus became more important, and the work I had done and the work my advisor had done became more important. And so after that, I went to NIH for 1 year to do a postdoc. on multiple sclerosis. And then because I was married at that point and had a 2-year-old, I had my first child in graduate school. Everybody thought I was leaving. I'd walk around pregnant. Everybody would say, "Oh, you're dropping out of graduate school." I don't think that would happen today, but this was many, many years ago. And I didn't. Obviously, I went on and I did my postdoc. And then my husband got a position, a faculty position at Mount Sinai Medical School, and we had to move. And that was the first time that moving was a good thing, because I had to drop my postdoc, which would have been 3 or 4 years, and go find another lab to work in. And I wound up working in Richard Axel's lab at Columbia University, all the way back to my father's alma mater, and had a really great time there, and did a lot of work on a variety of things, but mostly genetics and neuro stuff. And I was around to watch Linda Buck and my mentor, Richard Axel, win the Nobel Prize, or do the work that won the Nobel Prize. The best thing about being in a lab where people went on to win the Nobel Prize was the level of rigor. So the next thing I tell you is, besides being passionate and try to fail at something, is go somewhere where it's really hard. Don't pick anything that's easy. It was so much more useful and intriguing and stimulating to be in a lab where everybody was as smart or smarter than me, and everybody asked questions all the time. And it's held me in good stead to be. I spent 5 years in Richard's lab, and every time you got up to speak, it was like going to the Spanish Inquisition. There were just question, question, question. Everything you said was questioned. You really had to have thought through what you were doing. And that, even more importantly than what work you did, was the real training. And then after that, I was going to go into academics like everybody I knew around me, and I really went into drug discovery by accident. At that point in time, I was in New York City, and Merck was across the river in New Jersey. And this was 1989, and they had just come to the conclusion, and this was kind of across the pharma industry, that it's the genetic revolution. We should understand what our drugs are targeting genomically. We should understand, we should clone the targets, we should know what they are, and we should know exactly what the drugs are doing and how they're doing it. And to do that, we need to hire molecular genetics people, which I was one of. And so I got hired in this huge expansion at Merck, where there was tremendous enthusiasm from the top down to understand genes and their functions and find new ways to make drugs that way. And as a result of that, I spent 5 years on a project which we didn't realize at the time would turn out to be the cloning and the characterization of the first caspase. And for those of you who know what caspases are, they're a set of proteases, of enzymes that basically engineer cell death. We didn't know that at the time. The first one we cloned, we were hoping, would be something that would activate the immune system by turning on interleukin-1. And we were devastated to find out that very, very closely related enzymes actually did a great job of killing cells, which actually killed the program. But it was amazing, and it's a really good lesson about research. Caspases are very important in cells, and lots have been understood as a result of figuring out about them, but they were really first cloned by accident. And work that was done by other people in academics, not in the drug industry, were the ones who really nailed down their true function or their important function in apoptosis. And so after I left Merck, I decided that I wanted to do more drug discovery and less basic research, and I wound up in biotech. And so I went to a whole series of biotech companies. But again, it was kind of random. To be honest, at the time, we were living in Manhattan, and we were running out of money. We had 2 kids by then, and not enough money to live the way we wanted to and put them in the schools we wanted to in New York City, because my husband and I didn't make a whole lot of money as researchers in the sort of entry level of the drug industry. And biotech looked really exciting. The other reason I left is because it became clear at Merck and other pharmaceutical companies that the old school, where they had a small collection of small molecules, of chemicals that could be used to screen to make drugs, that was going to go away, and that there were now really fast ways to make all kinds of interesting libraries of new compounds by parallel synthesis, by combinatorial chemistry, and therefore you could now make these libraries very cheaply and have them all of a sudden. Before that, they had been accrued slowly over like 100 years from companies like Merck that had come out of the German dye industry and had a collection of molecules that were proprietary. And I realized then, which I give myself credit for, that a lot of the research was going to get diversified into small companies because there was nothing holding them anymore at the pharmaceutical companies. And it was also true at that time that biotech was doing new things like biologics and antibodies and not just small molecules. But I've never done anything but small molecules, and I just felt that they were going to go into the biotech realm where people moved quickly, made decisions, and took a lot of risks very quickly, and that small molecules could be there along with biologics and antibodies, and I wanted to be there. And I'll tell you, I never looked back, because when I was at Merck, I certainly had a good personal reputation. A lot of people knew me, but they knew me as someone who would raise their hand and ask a lot of questions, like, why are we doing it this way? And why can't we do it a different way? And I don't really understand why it has to be the way it was before. And those are not the kind of comments that give you a super long career in big pharma, unless you learn how to not ask those questions anymore. And it's really by personality. So my next thing is, after passion and all that, just know who you are. You should be in the environment where you can be your happiest. I can't tell anybody what that environment is. I found it by accident. I wished I'd found it sooner, and I wish I had figured it out better, because it took me years of wandering around to come to the right place. But the right place was pretty clearly biotech. It's very, very small. I've never been in a company bigger than 150 people since I left Merck. I've been in companies as small as 10 people. I went up the management ranks because I understood the science, but more importantly, I wanted to connect it to other things, like the business world and the commercial market for drugs. And I realized that I really was an applied biologist. At the end of the day, after I read 5 million papers, I loved doing that, I wanted to extract the information and I wanted to apply it to something. Not every researcher feels that way. Some people just want to answer questions. And for those people, you should stay in basic research. But for me, it was more important to make a drug that impacted somebody's life than it was to be somebody who comes up with a novel picture of the biological world. And so I stopped publishing papers and I went into biotech more about making drugs. And I learned how to make drugs in very small environments, and that's another whole story. And after I had done that for a while, by accident, I got downsized out of one company. So in biotech, you get fired a lot. I've been fired twice. Most people twice at least. You just move on. You get laid off. Things are very fluid. And because of that, I had an opportunity to start a company. So I was recruited by a couple of academic founders of a company, along with my second mentor, a person named Phil Whitcomb, who was actually at Amgen many, many years ago when it was run by George Rathman. And he was by then a starter of companies, kind of retired into being a counselor. And he got together with these two academic founders, and they recruited me to run the science in what became the company that David just mentioned, Proteolix. We actually brought in work from Yale and from Caltech and pursued a couple of targets that have to do with controlling protein degradation and turnover in cells. And we wound up coming up with an inhibitor that inhibits the proteasome, which is the structure in cells that just turns proteins over. And very fortuitously, somebody else had figured out that that was something that was very useful for the treatment of a particular cancer, multiple myeloma. And so we knew we were making another drug similar to one that was already on the market, but that had better properties. It was safer and more efficacious. And we went into the clinic with that drug, and we tested it in people. And I'll never forget the day when the data came across the fax machine. This was 10 years ago, probably the last fax I ever saw. And it was the data from a patient who had responded to our drug. And everybody popped the champagne, because I can't tell you what it feels like. It's just amazing to know that you were part of a team, or in my case, I led a team that made a drug that went into people that actually had an impact on somebody. But a couple of years later, I actually got to meet one of the people who was alive because of our drug. And so if you had to say to me, what was the pinnacle of your career, it was really that moment, because this person told us his story. He had multiple myeloma. He'd had it for several years. It's a disease of the bone marrow. It's an early or a late stage B cell, so it makes antibodies, and it gets stuck and overgrows in your bone marrow, and it breaks your bones. And it's very painful. And he had multiple lesions, and he went in on a stretcher to the hospital to go to our clinical trial because he was on his last legs and had taken all of the drugs that were out there, and he couldn't walk, which that's why he was on a stretcher. And he took our drug, and it was a very fast response, as was for other patients. And he was functioning a few months later, and he showed up to do a 3-mile walk run to raise money for multiple myeloma in San Francisco, and that's how we got to meet him, because normally you wouldn't meet the patients on your trials. And there he was, and he did the 3-mile walk, standing up and pain-free. It was just amazing to see that. And so for those of you who are thinking, why would I ever go into a career in the drug industry, I would tell you that if that kind of story makes you really excited, that's why you should go in, because you can have an impact in the world. So you can have an impact in the biological knowledge base and do great research, and that's very, very satisfying. Or you can do what I chose to do. I think they're both great paths. And I like, again, being in biotech because I have impact, and I like it because I spend my time communicating, which is why I come to things like this. I like to talk to people about science, and I like to talk to people about my experiences, and that makes me very happy too. Being the CEO of something, by the way, was nothing that was ever in my goals. If somebody had told me I was going to be a CEO, I would have laughed. I started out not having any interest in the business world, and I wound up so far raising $180 million of venture capital money in order to pursue what we want to do in these companies. And that's your job as the CEO, is to oversee things, be the person who has a goal and a vision, and inspire other people to follow you and raise the money. That's really the job, and communicate the passion. And it's a great job. I love it. But I wouldn't say it's what you have to do to be happy. I would say you have to do whatever makes you feel really, really great. Phil Whitcomb, the person who helped me start Proteolix, was diagnosed with prostate cancer about 6 months after we started the company. And unlike a lot of prostate cancer patients, he had a super aggressive form of the disease, and he didn't make it. I think he lasted about 18 months from diagnosis, and then he passed away from prostate cancer. And it devastated me because he was the second person who believed in me, and he's the one who talked me into being a CEO, which I'm very grateful for. It's been an incredible 10 years. And we named our drug, that's the drug that's now at Onyx, for Phil. His name was Phil Whitcomb, and the name of the drug is carfilzomib. And Car is for Carla, his wife, who passed away before I met Phil from another disease, an autoimmune disease called lupus. And we actually wound up developing a drug at Proteolix that also worked against lupus and other autoimmune diseases. So pretty weird how that works out. And it's because of people like my father and Phil who believed in me and who turned me on to the excitement of creating things and doing things scientifically, that it really worked out for me. And so my last piece of advice is find somebody probably older than you who really believes in you. It can be your parent, it can be a teacher, it can be somebody else that you meet in your career, and learn all you can from them. And if they really believe in you, make the best of that relationship, give back in that relationship, because that's really the emotional grease that really moves everything forward. Thank you.